Hi everyone, I'm delighted to open the proceedings at today's Proteomics World event. My name is Chris Whelan. I'm a director of Neuroscience Data Science at Johnson & Johnson Innovative Medicine. I also chair the UK Biobank Pharma Proteomics project. I'd like to start by acknowledging that it's an exceptionally exciting time to work in the field of proteomics. This is a rapidly advancing field, and as you'll hopefully hear from the speakers that follow me, it holds huge potential for precision medicine. So the potential of proteomics for precision medicine will be the primary theme of my talk today. I'd like to start by turning the clock back almost a quarter century to a pivotal moment for another related field, which is genomics. So in June of the year 2000, the completion of the Human Genome Project was announced, and this was met with some very valid fanfare. There were simultaneous telecasts from Bill Clinton and Tony Blair and Francis Collins, and this was heralded as the first great technological triumph of the 21st century. Francis Collins put it very beautifully by saying that we'd caught the first glimpse of our own instruction book, previously known only to God. And one of the central themes that was interwoven throughout all three telecasts was the potential of the human genome to usher in a new era of precision medicine, an era where we would have cures for diseases like Alzheimer's and Parkinson's and different cancers. So we're now almost a quarter century removed from these announcements. And it begs the question, are we in that era yet? And the answer is a little bit more complicated. If we look at the preceding 25 years, the official completion of the Human Genome Project was actually 2003. And in the years that followed, we entered what I might call an infamous candidate gene era, where we were exploring the differences in frequencies of a small number of gene variants in small numbers of cases and controls. So this was a hypothesis-driven approach, which ultimately was not particularly successful. Most of the findings could not really be replicated. And so the field changed tack towards the mid to late 2000s and took a more hypothesis-free approach, which is, of course, the genome-wide association study approach, screening from chromosomes 1 through 23 across much larger numbers of cases and controls. That approach proved to be a lot more successful, producing results that could be replicated, and so it grew in popularity, and we're now seeing population scale GWAS. This has followed us through to the present day, where it's now commonplace to see GWASs with hundreds of thousands of cases, and in some cases, millions of controls. In fact, this population scale approach is so popular that we've probably started to enter a new era, which I'd call the post-GWAS era, where we're trying to make sense of these thousands and thousands of genetic associations and trying to turn them into new drug programs and into new precision medicine approaches. So ultimately, genomics has held a lot of promise and delivered on some of its promises for precision medicine. But despite all that, precision medicine is still a little bit of a buzzword. So why is that? Why is it still a buzzword despite the promise and despite some of these successes? Well, I argue that it's probably because we've relied too much on genomics by itself, genomics in isolation. If you break precision medicine down into its fundamental components, ultimately, it's about developing the right drug for the right patient at the right time. And if we think about that in the context of genomics in isolation, finding the right drug can be very challenging. Of course, there are gene therapies, but gene therapies represent a very small proportion of the drugs that we develop. Most of our drugs are small molecules targeting proteins. Finding the right patient can also be difficult. Of course, if you're dealing with a rare disease, a disease that has Mendelian inheritance, then genomics is an incredibly powerful tool. But when it comes to more complex, commonly occurring diseases, things like polygenic risk scores still have unproven utility as clinical tools. And of course, then finally, right time. It can be challenging to use genomics to find patients at the right time because DNA is static. It doesn't change over time. And all of that stands in stark contrast to proteomics. With proteomics, I believe that we can find the right drug because most of our drugs target proteins. I think we'll have more success finding the right patients because there are already many examples in clinical practices of measuring proteins to diagnose and to manage conditions. Take, for example, ApoB for hypercholesterolemia. And then finally, I think that proteomics will prove very useful to find patients at the right time because proteins change over time. They change in response to differences in disease states. So to summarize this slide in a much more eloquent way, I'm going to steal an expression from my friend Melissa Miller and her colleague Eric Fahman at Pfizer, who say that proteins speak louder than genes. So ultimately, the protein could be a very, very powerful tool for precision medicine. And the rest of my talk, I'd like to focus on how we can reach that promised land of precision medicine with proteomics. And I'm going to argue that we can do that by continuing to support or to start to support three different subfields of proteomics. The first is population proteomics. That's already quite mature at this point. The second is clinical proteomics, which is not quite as mature, but it's starting to see some promise. And then the third would be translational proteomics, which is still very, very early. So let's just start by talking about that most mature field, which is population proteomics. And by population proteomics, of course, I'm referring to the measurement of thousands of proteins across tens of thousands of samples, reaching sort of population scale. Now, we are standing on the shoulders of many giants for population proteomics. There have been many groups that have applied multiplex proteomics on not quite tens of thousands, but have provided a foundation. So these are groups like the ARIC cohort, Interval, EPIC Norfolk, the Scallop Consortium. But I would say that the first group to truly do population proteomics across tens and tens of thousands of individuals were probably the Decode group in Iceland, led by Kari Stephenson, who you're going to hear from later today. So they performed aptamer-based proteomics across over 35,000 people in Iceland, and they've published many papers on this topic, probably the most widely cited being their protein GWAS, where they identified over 18,000 genetic associations with almost 5,000 proteins measured using an aptamer-based approach. But of course, despite the incredible sort of trailblazing of this study, this data set is not open access. So that led us to come together and pool our resources together and build a consortium of pharmaceutical partners to fund the generation of an open access proteomics data set. And of course, this led to the Pharma Proteomics Project. So Pharma Proteomics Project, or PPP for short, was 13 companies, and we all came together to fund the generation of Olink Explore 3072 measurements across 54,219 UK Biobank participants in blood plasma. One of the things that we did together, we didn't just pool our resources, we also pooled a lot of brain power, a lot of resources to conduct statistical analyses of this data set and make those analyses available to the public. We published on this late last year in Nature. We conducted a GWAS on the proteins. We identified over 14,000 protein quantitative trait loci, or PQTLs, and we also published some basic health associations with the proteins as well. I'm not going to spend too long on the different facets of that paper. I'd like to talk about how those PQTLs have been used by other groups downstream of the paper, and also talk a little bit about some of the other use cases beyond proteogenomics. But first, let's focus on proteogenomics. These PQTLs that you see here on the left-hand side of the screen have been used by a lot of different groups to find causal associations between proteins and different diseases. They use a technique called Mendelian randomization, which is often compared to combining proteomic data and genetic data to sort of run almost like a virtual clinical trial, to oversimplify. And this approach has been used to find essentially drug targets that have higher likelihood of success. We know now from many different peer-reviewed publications that if your drug target has supporting evidence from human genetics, then it's more likely to be approved ultimately. And so many different groups, as I mentioned, have applied Mendelian randomization using the UKB proteomic data. This is just one example for type 2 diabetes. Jan et al. Found 47 different plasma proteins that had strong causal associations with type 2 diabetes. Some of these proteins identified potential repurposing opportunities for drugs that were already in development, like GCKR targeting drugs. Some implicated things like Agar inhibitors in diabetic complications. And this is just one example just for type 2 diabetes. As you can imagine, there are already a number of other preprints and peer-reviewed publications applying Mendelian randomization with the pQTLs that we identified to find causal links between proteins and diseases, and ultimately to try to turn those causal links into new drug programs. So internally at J&J, my colleague Nasser took a slightly different approach to find new drug targets, quite a creative approach, I believe, where he started by conducting a typical sort of traditional GWAS for Parkinson's disease susceptibility, PD risk. And you can see the Manhattan plot showing the results of that PD risk GWAS on the left-hand side of the screen. And unsurprisingly, we identified many of the known sort of usual suspects that influence Parkinson's disease risk. You can see, for example, LRRK2, you see TMEM175, and SNCA, the synuclein gene. But the next thing that Nasser did was ask, of these gene variants that influence Parkinson's disease, which are also pQTLs, and what proteins do they impact? And so Nasser found that these Parkinson's-linked gene variants also impact levels of 577 different proteins. Most of those associations were in trans, and they were enriched for different pathways that had strong relevance to Parkinson's disease, like cytokine-cytokine interactions, and of course, lysosomal biology. So then the next thing that Nasser did was build a protein-protein interaction network based on these Parkinson's-linked proteins. And unsurprisingly, he found that there were a number of hub proteins, highly interconnected proteins, that we know a lot about already in the context of Parkinson's disease. For example, once again, we saw LRRK2 as a hub protein. We see ApoE, we see a GBA. But what's interesting, you'll notice the proteins that are highlighted by the green circles are somewhat novel proteins that have not necessarily been flagged by Parkinson's disease GWASs in the past. And Nasser zoomed in on one of these proteins called galectin-3. So this is a particularly interesting protein because the genetic association that has previously been flagged for Parkinson's disease has not been at the galectin-3 gene, not at LGAL-S3. It's actually been at another gene called GCH1. But as you can see on the left-hand side of the plot, the gene variant that sits within GCH1 actually influences galectin-3 levels in blood plasma in a genotype-specific manner. Nasser further looked at single-cell expression data and found that, particularly in microglia, Parkinson's disease patients have increased levels, increased expression of galectin-3. So this is all by way of saying, by combining the PPP, pQTLs, these population scale data sets, Nasser was able to zone down on a number of potential new drug targets for Parkinson's disease. Now, beyond the proteogenomic approaches, the proteins in and of themselves are also very useful to find potential new biomarkers. You see an example on the left of potential biomarkers that we flagged for schizophrenia at J&J, and on the right, some potential biomarkers that we flagged for Parkinson's disease. And what's quite nice about these population scale data sets is that they allow us to generate new hypotheses around biomarkers that oftentimes end up getting validated as part of more clinically curated cohorts. Take, for example, Parkinson's disease. We see an increase in DDC, dopamine decarboxylase. Since we observed this in the UK Biobank data set, it has since been validated independently in a Swedish cohort by Oskar Hansen, where it's been upregulated across a number of Lewy body diseases, as you can see from the graphs on the left. So a very useful data set for biomarker discovery. But not just biomarker discovery. I would say that this data set's also very useful for biomarker curation and refining the biomarkers that we're digging into. As an example, my colleague Li Ping ran something called a protein PheWAS. So, the volcano plots that you saw on the previous slides are looking for all the proteins that are dysregulated in particular diseases. PheWAS reverses that, of course, and looks for all of the diseases that are associated with a particular protein. And as you'll notice from the slide, I've given two examples of biomarkers that are already candidates for neurodegeneration, and those include neurofilament light chain on the left and GFAP on the right. What's interesting about the UKB data set, this population scale data set, is that Li Ping's PheWAS found that neurofilament light chain associates with over 200 diseases in UKB at a very stringent statistical threshold, 5 by 10 to the minus 8. Most of those diseases are not neurological. Most of them involve kidney diseases, they involve heart disease, etc. We do see dementias in there, but for the most part, these are non-neurological conditions. And that stands in stark contrast to GFAP, which is associated with only 29 illnesses at the same level of statistical stringency. And most of those illnesses are neurological. So this is a very useful way of parsing out the pleiotropy of different proteins and helping us find more disease-specific candidate biomarkers, with GFAP being more neurodegeneration specific in blood than neurofilament might be. So I want to finish up this section just by highlighting my confidence that this field, this subfield of population proteomics, will continue to expand. I think it's rapidly expanding already. The 3 papers that we published last year have already been cited quite a bit and well covered by the international media. And I'm very, very confident that the UKB PPP 55,000 K project is not the first of these population scale data sets that you're going to see. There'll be many more to come over the coming years. So with that, I'd like to move on to talk about a different potential subfield of proteomics, and that's clinical proteomics. Simply put, the application of proteomics in a primary care setting or in a clinical trial setting. And staying on the theme of UK Biobank, we're also seeing some very promising evidence that proteomics can be used in a clinical setting. A very beautiful study recently published by Giulia Carrasco-Zanini and Claudia Langenberg in Nature Medicine. They took the UK Biobank proteomic data and built models to predict the 10-year incidence of a range of different diseases. And they found that for 67 of those diseases, a model that included as few as 5 proteins and as many as 20 proteins outperformed models that included basic clinical information. So you see the clinical model is denoted by the gray line on the bottom. The model that includes both clinical information and blood biochemistry is on the blue line. And then finally, the orange line signifies the proteomic model. And as you can hopefully appreciate, for diseases with unmet need like celiac disease, lymphoma, multiple myeloma, and motor neuron disease, ALS, the proteomic model significantly outperforms those clinical and blood biochemistry models, and by a detection rate increase, which has clinical meaningfulness as well. So this is all by way of saying these proteomic tools could genuinely hold promise for clinical settings and potentially for settings in primary care in the future. Another widely published example came from Guo et al. Late last year in Nature Aging. They took a similar approach where they combined proteins together to predict the incidence of, in this case, dementia. And they found a model that included about 10 proteins, had a pretty good AUC of about 0.87 for predicting whether someone would develop dementia later in life or not. And some of those proteins, as you can hopefully see, included neurofilament and GFAP, which we spoke about earlier. But I think that these data are not just useful for predicting incident diseases. I think they also have a lot of utility for prevalent diseases, diseases that have already been diagnosed, stratifying those diseases. So let's give an example. One example would be Alzheimer's disease. So I work in the neuroscience space, and it's critically important that we take these very biologically heterogeneous illnesses and try to stratify them into more biologically homogeneous illnesses to help us pair the right treatments to the right patients. And so you'll see some work from Betty Times on the screen. She did this. She clustered proteomic data from Alzheimer's disease cases in the EMIF cohort. And she found 3 major subtypes based on the underlying biology, based on the proteomics. One subtype associated with hyperplasticity, one with innate immune activation, and another that involved blood-brain barrier dysfunction and complement activation. And we've been able to validate many of these subtypes in-house at J&J, thanks to the work of my colleague Bart Smets. And we're not just looking at Alzheimer's disease, we also have some very encouraging findings for subtyping of major depressive disorder using proteomics. I'm quite excited about this particular approach because we don't usually think of major depressive disorder in terms of any underlying biology. We just think about it like depression. And so we took the UKB PPP data, we took almost 3,000 people who had a diagnosis of major depressive disorder in UK Biobank a prevalent diagnosis. So they had this diagnosis at the time of their blood draw. And we ran nonlinear clustering on the proteomic data and asked essentially, are there biological subtypes of major depressive disorder? And we found that there are 3 robust subtypes of major depressive disorder. Some of those subtypes may involve inflammatory proteins, others more involving synaptic and neuronal proteins. So we are actively following this up at J&J, and potentially we could build predictive algorithms to find these different MDD subtypes in the real world and potentially in our clinical trials and help stratify into those biological subtypes that better respond to our treatments. But the last point I'd like to make on this topic of clinical proteomics is ultimately that the utility of proteomics in the clinic, I think it'll only really be shown if we ourselves, as pharma researchers, start to apply proteomics on our clinical trials. So that's exactly what we're doing at J&J. This is a very high-level example that I'll give you. It's been heavily censored, unfortunately, but we ran proteomics on plasma samples from a very large phase 3 trial. We actually had 2 studies from a phase 3 trial. And we ran proteomics, including Olink Explore HT, on untreated samples and treated samples from that trial. And then my colleague Jan-Fei conducted regressions to basically ask which proteins are changed by the drug, so proteins that are dysregulated in the treatment group, but not in the placebo group. And can we zone down on proteins that are changed consistently across study 1 of the trial and study 2 of the trial? And hopefully what you can see both from the volcano plots on the bottom left, as well as the protein-protein interaction network on the right-hand side of the screen, is that we could identify many different proteins that are impacted in the treatment group, but not in the placebo group. Some of those proteins directly interact with the target of the drug, as you can see on the bottom right, and some others form a very large network involving many different biological pathways. And some of those pathways, their biological functions can actually help explain some of the sort of secondary or tertiary endpoints changes that we're seeing on a clinical level from these patients. So very exciting work, and I'm hoping that we can talk about this in a lot more detail over the next few months. So there's a lot of promise for clinical proteomics, and I do see it start to develop more quickly as a response to population proteomics. But one field that has not really developed yet, which I think would be very, very valuable, particularly for drug discovery, is translational proteomics. And simply put, that would be trying to align biosignatures from human fluids or human tissues with biosignatures from cell lines. So let's stick on the topic of neuroscience. Let's take the example of Alzheimer's disease. We're already conducting proteomic profiling of postmortem brains from people with diseases like Alzheimer's disease. If you can imagine a world where we're also conducting proteomics on cell lines, patient-derived microglia or neurons from the same conditions, we could also be conducting proteomic profiling on cell lines that have been screened with candidate compounds to treat Alzheimer's disease. This approach, where we're aligning the cell line proteomics with the human tissue or fluid proteomics, could help us find more specific biomarkers, more specific drug targets, and potential tool compounds that could help accelerate the drug development process. Now, unfortunately, as I mentioned at the beginning, this field has not really taken off yet. I think that the foundation or the substrate for clinical proteomics is absolutely there. For example, Carlos Cruchaga has published his work where he's already conducted aptamer-based proteomics on postmortem brain samples from his NIGHT ADRC cohort. We internally at J&J have also been applying Olink Proteomics on, as well as mass spec and aptamer proteomics on about 250 postmortem brains. So there is a lot of substrate there, but it's not yet paired with proteomics of cell lines. So that'll be the next big step. And you might be asking, what's stopping us from conducting cell line-based proteomics? Well, that leads into the final section, which are the potential pitfalls, potential barriers to us really making precision medicine happen vis-a-vis proteomics. And the first pitfall, which is completely directly related to translational proteomics, would be, of course, the cost. So it remains to be seen whether Moore's law will apply to proteomics. We all know that Moore's law has applied very well to genomics. In fact, the cost of sequencing has sort of outperformed or outpaced Moore's law, as you can see on the left-hand side of the screen. I'm not sure whether this can happen for proteomics. You can't amplify proteins like you can DNA. We are seeing steady decreases in the cost of proteomics, but to make proteomics a workhorse tool like genomics or like RNA-seq, the cost does need to come down. And in that context, in a context where the cost is lower, we can start to apply proteomics of cell lines and start doing very large compound screening exercises like the L1000 exercise using proteomics instead of RNA-seq. Another thing to consider is the pace of innovation. So I think that at the moment, there's still a focus in the world of biomarker development on univariate biomarkers, meaning measuring one protein at a time. Again, in the field of neuroscience, there's a lot of excitement around p-tau217 in blood plasma as a potential blood-based biomarker for Alzheimer's disease, but that's still capturing just one aspect of the underlying biology of Alzheimer's disease. We know that these diseases are very heterogeneous, so we need to move from this univariate way of thinking to a multivariate way of thinking, and we need to get our C-suites and our regulators behind that idea. So we need to make sure that those that have the most power to influence this field are not the slowest to innovate. And on the topic of p-tau217, of course, these incredibly enabling approaches like Olink that allow us to conduct proteomics at very, very large scale in high throughput do not currently capture post-translational modifications like p-tau217. So this is going to become a particular roadblock, I believe. I think that PTMs are going to be very critical, particularly in spaces like neuroscience, where our most powerful biomarkers have been PTMs. So we need to consider to what degree does blood plasma also reflect disease processes in disease-relevant organs. So again, in neuroscience, blood is not necessarily the most relevant organ. How much can we capture in blood that truly reflects what's going on in the brain? And then finally, I think the most critical point is that we need to be very careful not to ignore the patient's perspective. This, coming full circle back to genomics, is a lesson that we can learn from genomics. We can learn this lesson particularly from direct-to-consumer testing with genomics. We need to understand the accuracy of these prediction tools that I've spoken about earlier. We need to understand how they apply across different ancestries. Most of these, as you can imagine, have been applied in people of white European ancestry. And we also need to understand ethical implications around whether insurance companies can access these kinds of proteomic prediction tools. So lots of things to think about in the future. You could imagine a future where we don't just have genetic counselors, but also proteomic counselors. And with that, I'd like to bring it all together and finish up. Hopefully what you've been able to take away from this talk today is that proteomics is becoming an increasingly recognized tool for drug discovery. We are using proteomics to increase the nominations of drug targets that are fundamentally embedded with human evidence. We're using proteomics to help us make decisions for existing drug programs, provide biological insights for existing programs. And then finally, and perhaps most excitingly, we're using proteomics to find new biomarkers and potentially build more clinical tools. I think that at the moment, while proteomics is increasingly seen as an important tool for drug discovery, it's not yet seen as a critical tool. I think it is a critical tool. If we want pharma to see it as a more critical tool, a couple of things need to happen. First, the price needs to fall. Second, our regulators and our R&D heads need to embrace multiplex ways of thinking rather than univariate ways of thinking. And then finally, we need to continue to advocate for collaboration between academia and industry, as we saw from the UKB project, but also collaboration with regulators. This is a highly collaborative field. We need to continue to embrace collaboration. And in that spirit, there's a lot of people to thank. It doesn't just take a village. It takes a small city to make these kinds of projects happen. So huge thanks to everyone. Thank you for inviting me today to Olink, and I hope everyone enjoys the rest of the meeting. Thank you.